group. So, or as the compounds that yield these substances upon the hydrolysis. So when coming to the general classification of the carbohydrates, these carbohydrates are generally classified into mono, di, oligo, and polysaccharide, which is depending upon their saccharin groups. So how many saccharin units we have, depending upon that, we are going to classifying the carbohydrates into four groups. Those were monosaccharide, disaccharide, oligosaccharide, and polysaccharide. The term saccharide or saccharide is coined for some carbohydrates like sucrose, which is presented in the cane sugar, glucose, fructose. Because of their nature of sweetness, the crystal structure and the solubility, depending upon the characteristics, we are classified the carbohydrates into these groups. And when coming to the monosaccharide, mono means one, only single. So these are often called as simple sugars. So which cannot be further divided or hydrolyzed. These are the basic units of carbohydrates, which is depending upon their number of carbon atoms. Here the carbon atoms are single, so that it again not going to split it into two halves. They may be subdivided into trioses, tetroses, pentoses, hexoses, or heptoses, or aldoses, ketoses, depending upon their aldehyde and ketone group presented. Here you can see the aldoses and ketone. Ketone group means double bond O. Wherever the glucose molecule containing the double bond O, that is ketoses. CHO, those were aldoses. Here triose, pentose, tetros, hexose. Hexose means 6, pentose means 5, Tetrose means 4, triose means 3 sugared molecules. Here you can see the structure of the molecules like aldose, ketose, glucose and fructose. The most important hexo sugar is glucose, which is an ultimate energy resource. The structure of glucose is given as the straight chain, the naturally occurring form. Here you can see the structure of glucose and aldohexose. So when coming to the monosaccharides, when coming to the disaccharides, name itself indicating like two molecules of saccharin molecules. What two molecules here? These yield two molecules of same different monosaccharides when it get hydrolyzed. So those were like sucrose and the sugar which is presented in the milk, that is lactose and maltose. For example, upon the hydrolysis, sucrose molecules can give rise to glucose and fructose, where as maltose gives two molecules of glucose. So the maltose is the combination of two glucose molecules. When coming to the oligosaccharides, these yield 3 to 6 monosaccharides units on hydrolysis. Those were raffinose and verbenose. And polysaccharides, these are the complex carbohydrates, often insoluble, and which is made up of a long chain monosaccharide units. So here there are a combination of monosaccharide subunits. Means n number of, poly means many. So n number of saccharin molecules together called as polysaccharides. This gives more than 6 monosaccharide upon hydrolysis. These may be of homopolysaccharide. Homo means same, similar. So similar type of monosaccharides together forms into a group of chain called as polymonosaccharide, um, homopolysaccharide, and hetero means different. So here the presence of different types of monosaccharide subunits you can see. So the example for the heteropolysaccharide is galactosamine and homopolysaccharide is glycogen. Here you can see the structure of the starch and chitin. The general examples for the polysaccharide is starch, cellulose, dextrin, glycogen. All these were homopolysaccharides, and glycosamine, galactosamine are the heteropolysaccharide. Mainly, the main source of polysaccharides are which presented in the rice. That's why in these days people are most of the people are stopped eating the best source of carbohydrate called rice. Uh, People are mostly focusing on the alternatives for the carbohydrates to avoid to avoid the diabetes, and wheat and other cereals and roots, stems and tubers of some plant materials. This is all about the structure and the qualities of carbohydrates. When coming to the functions, what is the function of carbohydrates? Yes, as we discussed in the first few lines, almost all the animals which utilizes the carbohydrates as the respiratory fuels. So we need carbohydrates as an energy source. This is the source which is useful for the energy production. And carbohydrates in the form of glucose and glycogen serve as a main source of energy for all the day by day activities. That is what carbohydrates considers as a source of energy. And carbohydrates which plays an important role in metabolic processes, those were gluconeogenesis by supplying carbohydrates to form amino acids as well as glycerol moiety of the fats. And the other function is it improves the uh, roughage, which is called as indigestible part of the carbohydrate diet. For example, cellulose of fruit, vegetable salads, and wheat husk. It gives bulk to the diet and helps to satisfy the appetite. The roughage stimulates the peristalsis and bowel movement. When sufficient amount of carbohydrates that provided in the diet and proteins are not at all utilized for the energy purposes, the as a result of uh, this one, the protein is utilized for building the tissues. This is known as protein sparing action of carbohydrate. After all, the main wording, yes, carbohydrates form the main source of reserved foods such as glycogen, starch. So here we can say like definitely carbohydrates plays the major role for the energy production of any animal body. When coming to the metabolism, animals gets carbohydrates through the food. After digestion, they will absorb into blood circulation as a glucose, and finally it reaches tissues. In tissues, the oxidation of glucose yields energy rich substances like ATP. Here the energy yield substance. What is the energy yield substance here is ATP. ATP means adenosine triphosphate. And during the process of oxidation, the glucose molecules is first converted into pyruvic acid through the uh, chain of glycolysis. In the presence of oxygen, the pyruvic acid is further broken down into the CO2 and water by way of citric acid. That is that happens in mitochondria during the cycle of Krebs cycle. The glucose can be stored in the form of glycogen in liver and muscle, that is converted to fat in the adipose tissue, that is fatty tissue. Because of glucose in the body, uh, which is the preferred source for synthesizing the ATP. It uses, depending upon the needs of the body cells, the fate of glucose happens like this, like ATP production. Here, ATP production by the body cells, which require the immediate energy. Then glucose is oxidized to produce ATP. And glucose is not needed for immediate ATP production, can enter one of the several orders metabolic pathways, and then the amino acid synthesis happens. The cells throughout the body can use glucose to form several amino acids, which then can be incorporated into proteins, the amino acids uh, incorporated into proteins. And then the next step is synthesis of glycogen. The glycogen synthesis happens in the liver tissue, hepatocytes and muscle fibers, that those can perform the uh, cycle like glycogenesis, in which a couple of like 100 of glucose monomers are combined to form the polysaccharide glycogen. The total capacity of glycogen storage happens is about 125 grams in the liver and 375 grams in skeletal muscle. Triglycerides synthesis, where the glycogen storage areas are filled up, hepatocytes can transform the glucose to glycerol, and fatty acids that can be used for lipogenesis, lipid formation. This is for the synthesis of triglycerides, which are deposited in adipose tissue, which has the unlimited storage capacity. Then the oxidation of glucose to produce the ATP, adenosine triphosphate, is also called as a cellular respiration, which are called catabolic activities. Cat means degradation. So here the what is going to degrade here is glucose. Here the glucose is 6-molecule compound carbohydrate. The splitting of 6-molecule compound into the ATP, 2 molecules of ATP production takes place. That is what it is considered like a macromolecule into micromolecule. That is what catabolic activities happens with the cycles of glycolysis and acetyl coenzyme formation and Krebs reaction. So what is glycolysis here? Yes, it is the set of reactions uh, where one glucose molecule is going to oxidize and to the formation of 2 molecules of pyruvate. The reaction are produced 2 molecules of ATP and 2 energy containing NADH2, because glycolysis does not require any oxygen. So it is a way to produce ATP So called as since it will not require any oxygen molecules for the process, so called it as a anaerobic cellular respiration, which is happening anaerobically. So formation of acetyl coenzyme A, this is a transition step that prepares pyruvate for entrance into the Krebs cycle and also produces energy containing NADH plus H plus, and also along with CO2 also releases. So for this one, this is called as a transition step, uh, which prepares the pyruvate for entering into the Krebs cycle. Where the pyruvate came? Pyruvate came from the glucose, glycolysis. There, one glucose molecule is oxidized into two molecules of pyruvate. So that pyruvate is going to entering into the another cycle called Krebs cycle. So here it is the preparation step, such as acetyl-CoA formation, acetyl coenzyme A formation. So after this transition step, the pyruvate is going to entering into the Krebs cycle, where acetyl coenzyme A is going to oxidize, and the oxidized when it is going to oxidize, the releasing of CO2 taken place along with energy and ATP. Here the energy containing molecules are the NADH plus H plus, means NADH2 along with FADH2. So the next step is electron transport chain. Name itself indicating that electron transport chain. It oxidizes NADH2 plus FADH2 and transfer electron through a series of electron carriers. The Krebs cycle electron carriers, the one which carries the electron, and the Krebs cycle and the electron transport chain both require oxygen to produce ATP, and which is collectively called as aerobic cellular respiration. Name itself indicating that it requires the oxygen. So if since it requires the oxygen, we consider it as a cellular aerobic cellular respiration. So the next level is gluconeogenesis, name itself indicating that gluconeogenesis means what is happening here is which is happening in the liver. So when the liver storage is low of glycogen and the body starts catabolizing triglycerides and proteins. The glycerol part of the triglycerides, lactic acid, and some certain amino acids can be converted in the liver to glucose. The process of forming glucose from one carbohydrate source is called as gluconeogenesis. So finally, the process of forming glucose from non-carbohydrate sources called as gluconeogenesis. This is happened when the human is doing the fasting. Uh, when the when people do fasting, definitely this is what happened. Gluconeogenesis happened. What happened in the gluconeogenesis means here the formation of glucose taken place from the non-carbohydrate sources. If the carbohydrate is not readily available, so body utilizes the try to synthesize the non-carbohydrate compounds into carbohydrate like glucose. So besides the glucose involved most of the catabolic reactions to generate ATP. Glucose, which takes part in other several reactions called anabolism. Ana means the um, small molecules into the bigger molecules, where we can listen all these words like mm, catabolism is the example. The example for the catabolism is respiration. The example for the anabolism is photosynthesis, where in the photosynthesis, where the small molecules group together to form a bigger molecule called glucose. So there the activity of CO2 molecules taken place, those were smaller molecules. The formation of bigger molecule taken place. Up to now we discussed about the respiration process. There the bigger molecules like glucose is split down into the smaller molecules called the single carbon molecules called ATP, and the what we call in the Krebs cycle or and glycolysis, we can see the production of pyruvate. So in case of anabolic reactions, anabolism means a group down of smaller molecules into bigger molecules. So what is happen in this case? See, glycogenesis. So name itself, if glucose is not there in the blood, not there in the body, just now we discussed about the liver. So if glucose is not needed, if glucose is not at all needed immediately for the ATP production, it combines with the many other molecules of the glucose to form the glycogen molecules. A polysaccharide, that is the only stored form of carbohydrate in our body. The insulin of the pancreatic beta cells that stimulates hepatocytes and skeletal muscle cells to carry out glycogenesis. The body can store about 500 grams of glycogen and 75% in skeletal muscle fibers and rest in the liver cells. So here you can see 500 grams means 500 grams of glycogens, you can see in that 75% in the skeletal muscle fibers and rest in the liver cells. So when coming to the glycogenolysis, here what happens here means glycogenolysis, when body requires high amount of the ATP energy and the glycogen which is stored in the liver, that is broken down into the glucose and released into the blood cells to reach the cells for the catabolic processes. So the total process of forming glucose from glycogenesis is called as glycogenolysis. So when coming to the glycolysis means just now we discussed the conversion of macromolecule into micromolecules. Here the macromolecule is glucose. The broken down of the macromolecule into smaller molecules like pyruvate happens here. We'll see. Glycolysis is the metabolic pathway. Which metabolic pathway it is? Catabolic. So that converts glucose C6H12O6 into pyruvate called CH3COO minus and CH3COOH, which is a hydrogen ion. The freely energy released in the process is used to form the high energy molecules. Here the along with the pyruvate, we also can see the presence of ATP, adenosine triphosphate, NADH, reduced nicotinamide, adenine dinucleotide. Here the glycolysis is a sequence of 10 enzyme involved, 10 enzyme catalyzed reaction. Mostly monosaccharide, those were fructose, galactose, can be converted to one of these intermediates. The intermediates may also be directly useful rather than just utilized as steps in the overall reaction. For example, the intermediate uh, molecule like dihydroxyacetone phosphate DHAP is the source of glycerol that combines with the fatty acids to form the fat. So here glycolysis is the oxygen dependent metabolic pathway. The wide occurrence of glycolysis that indicates it is an ancient metabolic pathway. Since why means it is every day it is happening in either way, like in a human body or plant body, wherever you wanted to see there the glycolysis happen. If someone wants to lift their hand also, that energy reaction may have to perform because we need energy production. So the it is occur worldwide occurrence of glycolysis that indicates it is an ancient metabolic pathway. Along with that, glycolysis is found in the great majority of organisms everywhere which is alive today. In organisms that perform cellular respiration, and glycolysis is the first stage of the process. In most of the organisms, glycolysis occur in cytosol. And the most common type of glycolysis is EMB pathway. So here the EMP stands for Emden Meyerhof Parnas pathway, which was discovered by the scientist Emden. His Gustav Emden and Otto Meyerhof and Jakob Karol Parnas. Glycolysis are refers to the other pathways. Those were entered Diodov pathway, ED pathway, and various heterofermentative and homofermentative pathways. Um, Here, the glycolysis is of like preparatory pathway we can see, and payoff pathway. Okay, here which consisting of mostly 4 steps. Here the 4 steps you can see here is one is energy requiring pathway. Name itself indicating it requires the energy for doing the pathway. So in this phase, the starting molecule of glucose gets arranged, like rearranged, and 2 phosphate groups are attached to it. The phosphate groups makes the modified sugar now called fructose 1,6 biphosphate, which is unstable one, which is splitted into half and forms into 2 phosphate bearing 3 carbon sugars. That is energy requiring phase. Energy investment phase. Here the energy investment phase is glucose is first converted to fructose 1,6 biphosphate in a series of few steps that use 2 ATP molecules, which requires the 2 ATP molecules. Then unstable glucose 1,6 diphosphate that in where we discussed in the energy requiring phase, that split down into 2 carbon molecule called DHAP along with glyceraldehyde 3-phosphate. Here the glyceraldehyde 3-phosphate can continue with the next steps of the glucose glycolysis, and DHAP that is dihydroxyacetone phosphate can be readily converted into glyceraldehyde 3-phosphate. So energy payoff phase. Here this is happened in a few steps which produces the NADH and 2 ATP molecules, where the glyceraldehyde 3-phosphate molecule is converted into pyruvate molecule. This is happens twice for each molecule of the glucose. since glucose is split down into uh, 2 3 carbon molecules, both of which that will go through the final steps of the pathway. And 3 carbon sugars formed when the unstable sugar breakdown. Uh, so here the formation of the 2 molecules of glyceraldehyde 3-phosphate happens. Only one glyceraldehyde 3-phosphate is entering into the next step. So the last step here is energy releasing pathway or energy releasing phase, where you can see in this phase the 3 carbon sugar, uh, that is glyceraldehyde 3-phosphate, is converted into the pyruvate. This is the pyruvate is the final compound of the glycolysis. Here you can see the formation of uh, the 1,3-bis phosphoglyceraldehyde. Here the 2 ATP molecules is going to releasing from the 2 ADP. Here it is the first ATP formation reaction. This is called a substrate level phosphorylation. And here the addition of phosphorylation means releasing of phosphate taken place. So here one phosphate molecule going and 3-phosphoglycerate formation taken place. Here 2 molecules of phosphoglycerate is formed where hydrolysis happened for the formation of phosphoenol pyruvate. Here the 2 molecules of phosphoenol pyruvate formation taken place. So second ATP formation step here. This is also a substrate level photophosphorylation. Here the about 10 involvement of 10 enzymes taken place. Those were hexokinase. Here in the first step, glucose is going to converted into glucose 6 phosphate, where the utilization of energy molecules taken place. So the ATP is converted into ATP. So this particular step is called as priming reaction. Why it is called as priming reaction means where the conversion of glucose is taken place into the glucose 6 phosphate with the activity of hexokinase enzyme. This is called as preparatory phase up to the formation of glyceraldehyde 3-phosphate along with DHAP. So the next enzyme is hexoisomerase. With the activity of the hexoisomerase, the glucose 6 phosphate is going to converted into fructose 6 phosphate. Then this reaction happens with the activity of phosphohexoisomerase. So the second priming reaction is conversion of fructose 6 phosphate into fructose 1,6 biphosphate by the activity of the phosphofructokinase enzyme. Here the phosphofructokinase enzyme plays the major role for the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Here the utilization of energy molecule taken place, ATP is converted into ADP. Here the utilization of energy taken place, one electron is going to loss. Then the cleavage of 6 carbon sugar. Here the cleavage of 6 carbon sugar taken place after the conversion of phosphate into 3 carbon sugar phosphate. Those were glyceraldehyde 3-phosphate. Here it is a 3 carbon sugar along with the formation of dihydroxyacetone phosphate. Here the activity of aldose enzyme taken place. So up to now we have seen the preparatory phase in the glycolysis. First priming phase and second priming reaction. Next is payoff phase. Here in case of payoff phase, the activity of glyceraldehyde 3 phosphate dehydrogenation. Here, the removal of hydrogen molecule taken place. What happens when the removal of hydrogen taken place? Here it is called as payoff phase. Why we are calling as a payoff phase here is oxidative conversion of glyceraldehyde 3 phosphate to the pyruvate taken place, and the coupled formation of ATP and NADPH also taken place. NADH also taken place. So in the first reaction, the formation of here the oxidation and phosphorylation, both the reactions happened here. So with that, the formation of 1,3 biphosphoglycerate, two molecules of 1,3 biphosphoglycerate formation taken place. First ATP formation reaction is called as is happened here, that is substrate level phosphorylation happened here with the activity of the phosphoglycerate kinase. So here two energy molecules releasing means 2 ATP is going to release. So next, the formation of 3 phosphoglycerate, two molecules of 3 phosphoglycerate formation taken place, and where the activity of phosphoglycerate mutase is converted into 2 phosphoglycerate, such as two molecules, and the other one is when uh, the enzyme called enolase, with the activity of the enolase, here the enolase dehydrolyze the reaction with the activity of the enolase, the two molecules of the water molecules releases, where the conversion of phosphoenol pyruvate, PEP, pepper, two molecules of pepper formation taken place. So second ATP formation reaction is substrate level, that is last reaction, where the involvement of pyruvate kinase enzyme, that pyruvate kinase enzyme converts the phosphoenol pyruvate into two molecules of pyruvate. This is what happens in the glycolysis. Here the glycolysis is split down into two reactions. One is preparatory phase, the other one is payoff phase. This is this is this is about glycolysis happens in the any living body. So here what is happening here? 6 carbon compound, that is 6 carbon sugar is converted into 3 carbon molecule called pyruvate with the series of reactions with the series of energy consuming and energy releasing reaction happened. The overall process is called as glycolysis. So finally, each reaction in glycolysis that is catalyzed by their own enzyme. The most important enzyme for the regulation of glycolysis is phosphofructokinase that catalyzes formation of unstable 2-phosphate sugar molecule fructose 1,6 bisphosphate. Fructose kinase which speed up the reaction or slow down the glycolysis in response to the energy needs of the cell. Overall, glycolysis converts 1,6 carbon molecule to glucose into 3 carbon molecules of pyruvate. So in one term, there are 10 steps reaction here as just we discussed. A phosphate group is transferred from ATP to glucose for making the glucose 6-phosphate. Glucose 6-phosphate is more active than the glucose. In addition to the phosphate that traps glucose inside the cell, since glucose with a phosphate that can't readily cross the membrane. As we discussed, it is happened in the cytosol. So glucose 6 phosphate is converted into its isomer called fructose 6 phosphate, where the transfer of phosphate group is transferred to the fructose 6 phosphate for the production of fructose 1,6 biphosphate, sorry, fructose 1,6 bisphosphate. This step is catalyzed by the activity of the enzyme called phosphofructokinase, which can be regulated or speed up or slow down the glycolysis reaction. The next step is fructose 1,6 biphosphate, which splits to the formation of 3 carbon compound sugar, that is DH AP, dihydroxyacetone phosphate, along with glyceraldehyde 3 phosphate. These are the isomers of each other, but only glyceraldehyde 3 phosphate that can directly enter into the next step of the glycolysis, where DHAP, the other molecule of the DHAP, that converted into the glyceraldehyde 3 phosphate. The two molecules exist in the equilibrium in the same way, but the equilibrium is pulled strongly downward in the scheme of the uh, as a glyceraldehyde phosphate that is used up. Thus, all the DHAP is eventually converted into glyceraldehyde 3 phosphate. Since second half of the glycolysis of the reaction should happen because of this reaction only. Otherwise, DHAP cannot enter into the next steps of the reaction. That is what, once the glyceraldehyde 3 phosphate is entering into the glycolysis, in the other hand, the conversion of the DHAP is DHAP into the glyceraldehyde 3 phosphate taken place. It is happened just like a cycle. The, the next step is glyceraldehyde 3 phosphate, which is converted into 1,3 bisphosphoglycerate. This is a redox reaction where you can see NAD plus is converted into NADH with the releasing of one hydrogen ion, which is an inorganic phosphate is also a reactant for this reaction, which is catalyzed by glyceraldehyde 3 phosphate dehydrogenase. And the next step is 1,3 bisphosphoglycerate is converted into 3 phosphoglycerate by phosphoglycerate kinase. This step converts an ADP into an ATP. ADP means di, adenosine diphosphate. After the addition of the H molecules, here you can see the energy molecule formation, that is ATP, adenosine triphosphate. Sorry, not H molecule, phosphate molecule. Here 2 molecules of phosphate molecule is converted into 3 molecules of energy molecule, that is ATP. And the next step is 3 phosphoglycerate is converted into 2 phosphoglycerate by phosphoglycerate mutase enzyme activity. And the next step, just above the last step, is the formation of phosphoenol pyruvate by the activity of enolase from 2-phosphoglycerate. Here, this reaction which releases the hydrogen, which releases the water molecule. And the last step is conversion of uh, phosphoenol pyruvate to pyruvate by pyruvate kinase enzyme activity, and ADP molecule, adenosine diphosphate molecule, is converted into ATP. The overall reaction is here. 2 ATP molecules are used in steps like 1, 2, 3. 2 molecules of uh, ATP molecules generated in 7th step, and 2 more molecules generated in step 10. So this gives about 4 ATP molecules produced, substrate the 2 ATP molecules used in the steps 1 to 3. from the 4 generated at the end of the step 10, which ended up with the net total amount of 2 ATP molecules is produced. If the oxygen available, the pyruvate can be broken down and oxidized all the way to produce the carbon dioxide in cellular respiration, making many molecules of the ATP. So this is how glycolysis happens. After this, the when the ATP and pyruvate formation taken place in the cytoplasm, cytosol, that is enter into the cytoplasm for the formation of the acetyl-CoA. Then the Krebs cycle happen in the mitochondrial matrix on the F1 molecules, where the oxidative photophosphorylation happened in the mitochondrial inner membrane. Where the energy molecule is released. The cellular respiration, like glycolysis, is the first pathway of cellular respiration, which oxidizes the glucose molecules. It is followed by the Krebs cycle, and where the oxidative photophosphorylation is after the oxidative photophosphorylation produces the ATP molecules. This is about the entire Krebs cycle, but up to, just we finished with glycolysis. Then what is happening in the Krebs cycle? We'll see. Once the formation of the pyruvate happens in the glycolysis, the pyruvate converted into acetyl-CoA in order to enter the citric acid cycle, where the acetyl-CoA enzyme activity where in the cytoplasm, the acetyl coenzyme with the activity of acetyl coenzyme activity, here the addition of carbon dioxide taken place, where the conversion of the acetyl CoA happens. So from that, the acetyl CoA is going to entering into the Krebs cycle. Here you can see the Krebs cycle which is happening in the mitochondrial matrix. So in case of mitochondrial matrix, in order to pyruvate the product of glycolysis to enter into the next pathway, it must undergo the several changes to become like a acetyl coenzyme. And here acetyl-CoA is a molecule that is further converted into the OAA. Here the OAA means oxaloacetate. Here you can see the OAA present. OAA acetyl oxaloacetic acid, which enters the citric acid cycle, that is also called as Krebs cycle. It is also in turn called as tricarboxylic acid cycle, that is TCA cycle. In another term, it is called as citric acid tricarboxylic carboxylic acid cycle and Krebs cycle, where the conversion of pyruvate to acetyl-CoA is the three-step process. The conversion of pyruvic acid to the acetyl-CoA, the citric acid taken place in the matrix of the mitochondria. Almost all of the enzymes of citric acid cycle, those were soluble with a single expression of the enzyme that is succinate dehydrogenase, which is embedded in the inner membrane of the mitochondria. Unlike glycolysis, the citric acid cycle is a closed loop one. Here you can see the cytosol where the pyruvate is presented. Then in the mitochondrial matrix, there is a 8-step of reaction you can see. In the first step, the last part of the pathway regenerates the compound used in the first step. It is like constitutive of 8 steps. A series of redox, dehydration, hydration, and decarboxylation means 4 that produce 2 carbon dioxide molecules along with 1 GTP or ATP, guanosine uh, triphosphate or adenine triphosphate and reduced form of NADH and FADH. This is considered as a aerobic pathway because the NADH and FADH2 produced must transfer their electrons to the next pathway in the system, which will use the oxygen. Because of that, it is considered as a anaerobic reaction. If this transfer do not occur, the oxidation steps of the citric acid cycle and do not uh, citric acid cycle produces very little ATP directly that do not directly consume the oxygen. The acetyl group from the acetyl CoA is attached to the 4 carbon molecule called OAA, oxaloacetic acid. That OAA is produced um, in the cytosol and to form a 6 carbon citrate molecule through a series of steps called the citrate is oxidized and releasing 2 carbon dioxide molecules for each acetyl group that fed in the cycle. In this process, 3 NAD plus molecules are reduced to form the NADH. One FAD molecule is also reduced to form FADH2 and one ATP and GTP is formed depending upon the type of the cell is produced through the way of substrate level phosphorylation because of the final product of citric acid is also the first reactant and the cycle runs continuously in the presence of sufficient reactants. So what is the first step here is called as a condensation step, which means the combination of 2 carbon acetyl group. But here the combination of 2 carbon acetyl group taken place. That is acetyl co uh, with the 4 carbon acetyl molecule called oxaloacetic acid OAA. Okay, this oxaloacetate molecule to form 6 carbon molecule called citrate. So CoA is bound to the sulfhydryl group and diffuses away for eventual combination with the another acetyl group. Here you can see all the reaction and this step is which is a reversible step from here to there and there to here you can see. This is also called as exorganic reaction. Means the X means outside, in endo means inside. So the releasing of energy molecule taken place. The rate of reaction is which is controlled by negative feedback and the amount of ATP available. If ATP levels are increasing, the rate of reaction decreases. Okay, because when the ATP molecule is more in the reaction, the releasing of ATP is happened. So then the rate of reaction decreases. If the ATP is in shorter supply, the rate of reaction increases. This is what happens and in the first step. When coming to the second step, citrate loses one water molecule and gains another. A citrate is converted into isomer, isocitrate. So here in the third and fourth steps, what is happened here means the production of succinyl coenzymes in between The production of alpha ketoglutarate also taken place. This is called as oxidation and decarboxylation steps. Name itself indicating decarboxylation happens here. The releasing of CO2 molecule taken place, and it which catalyzes the uh, succinyl CoA along with NADH. And the next step is a phosphate is substituted for coenzyme A, where high energy bond is formed. This energy is used in the substrate level phosphorylation, where the conversion of succinyl group to the succinate. So finally, what happens mean guanine triphosphate or ATP is releases. These two forms of the enzymes are also called as isoenzymes. Means the two are similar. For this step, depending upon the type of the animal's tissue in which they are performing the reaction, one form is found in tissues that use large amount of ATP, such as heart and skeletal muscle. In the heart muscle and the skeletal muscle, this forms produces ATP. The second form of the enzyme is found in tissues that have a high number of anabolic reaction. Those were liver. And this form of GTP means guanine triphosphate. GTP is energetically equivalent to the ATP. However, its use is more restricted in particular protein synthesis, primarily uses GTP. So depending upon the tissue, the performance of the ATP and GTP taken place. And the next step is the sixth step is called as dehydration. Name itself indicating dehydration, releasing of water molecule taken place. Here the conversion of succinate into the fumarate. Two hydrogen atoms are transferred to the FAD. The production of FADH2 taken place. The energy contained in electrons of these atoms in sufficient to reduce NAD but adequate to the reduced FAD. Unlike NADH, this carrier remains attached to the enzymes and transfers the electrons to electron transport chain directly, where the process is made up of by possible localization of the enzymes and catalyzing this step inside the inner membrane of the mitochondria. This is what the entire Krebs cycle looks like, where from the acetyl CoA to citrate, isocitrate, alpha ketoglutarate, and succinyl CoA and succinate, fumarate, malate. Finally, four carbon compound called oxaloacetate OAA production taken place, where you can see the releasing of energy molecules. Water is added to the fumarate during the step seventh, that is malate is produced. The last step in the citric acid cycle generates oxaloacetate, that is oxidizing malate. Another molecule of NADH is produced. What are the products of citric acid? Here you can see the two carbon uh, atoms comes into the citric acid from each acyl group, representing the 4 out of the 6 carbons of one glucose molecule, and 2 carbon dioxide molecules are released one each turn of the cycle, where however these do not necessarily containing the most recently added carbon dioxide atoms. The two acetyl carbon atoms will eventually releases on latter turns of the cycle. Thus, all 6 carbon atoms from original glucose molecules are eventually incorporated into the carbohydrate carbon dioxide. Each turn of the cycle forms 3 NADH molecules and 1 FAD molecules. These carriers will connect with the last portion of the aerobic respiration to produce as the ATP molecules. Here the ATP molecules are the energy molecules and one GTP and ATP is also made in the each cycle. several of the intermediate compounds in the citric acid cycles can be used in the synthesis of non-essential amino acids. Therefore, the cycle is amphibolic. Amphibolic means here the behavior of the catabolic and anabolic you can see in only one step. So after glycolysis, pyruvate is converted into the acetyl-CoA in order to enter the citric acid cycle. Okay. In the next class, we'll discuss about in the next class we will discuss about the next topic.